We're going to finish up signaling today. Just to remind you, we're talking about signal transduction, how we get information from outside the cell to inside the cell, resulting in a cellular response. We are going through these three basic pathways. We have made it through what a G protein coupled receptor is and sort of the generalities of that signaling pathway. We've made it through adenyl cyclase. We will talk about the phospholipase C pathway today. And actually, we'll talk about two different ways that that phospholipase C pathway can be activated. Just as a review, the adenyl cyclase. We activate the adenyl cyclase with the G protein that's been that's exchanged as GDP for GTP. That activates adenyl cyclase, which generates cyclic AMP, which activates protein kinase A. I'm last day jitters or something. I'm sad the class is ending. All right, so that's what we went over for adenyl cyclase pathway. Let's take a look at how this works for the phospholipase C pathway. All right. We're still talking about G protein pathways. So it's a very similar start. We have a GPCR, a G protein coupled receptor, that's going to bind some extracellular, so outside the cell ligand signal to activate it. That's going to promote GDP for GTP exchange in our G protein. That's going to allow the alpha subunit to dissociate from the beta gamma. That GTP alpha subunit then can interact with phospholipase C. Notice the location of all of this. Our GPCR spans the membrane. Our phospholipase C is embedded on the inside of that cellular membrane. That phospholipase C, when interacting with our alpha GTP, that then becomes active. And phospholipase C, when activated, interacts with PIP2, a phosphatidylinositol 4,5 bisphosphate, which is why we say PIP2. It's a lot easier to say. All right, so that PIP2 is partially embedded in the membrane, but then here is our 3-phosphate group. When activated by our alpha GTP, phospholipase C cleaves PIP2 into two molecules. One is this IP3, so our inositol 1,4,5 triphosphate, which then is released into the cytosol. And then we have this bit of that molecule that's left in the membrane. That should look familiar to you. What is it? All review for your final. What are those? Right? What do these zigzags represent? What kind of molecules are those?
Oh, yeah.
What are those two zigzag things connected to? What do we call it?
We drew this. It's two batches of back and yeah.
All right, everyone get their answers in?
19.
All right, the class thinks that it is diacylglycerol. That is correct. You guys have been studying. Good job. All right, so our activated phospholipase C splits PIP2 into our inositol triphosphate and a molecule of diacylglycerol. Remember diacylglycerol, two fatty acid chains bound to a glycerol molecule, all right, which is soluble in the membrane. Because phospholipase C has split this molecule in two, we have two molecules that can move on and interact with other proteins. IP3 goes on to interact with ion channels in the membrane of the endoplasmic reticulum. So ligand activated ion channels. Once IP3 binds to those ion channels, so DAG first. DAG interacts with protein kinase C. Protein kinase C is not fully activated yet. In order for that to happen, IP3 interacts with those ligand-gated ion channels in the membrane of the ER, causing those ion channels, which are calcium channels, to open and release calcium from the lumen of the ER into the cytosol. That calcium release then can have multiple effects. First of all, we need calcium as the second component to fully activate our protein kinase C. The protein kinase C is activated by the combination of our diacylglycerol plus calcium. And now that protein kinase C is capable of interacting with other proteins and phosphorylating them. Okay, so in the phospholipase C pathway, we see the cleaving of PIP2 into DAG and IP3. Each one of those goes on to interact with additional proteins. IP3 releasing calcium, which in combination with DAG activates protein kinase C. And then that protein kinase C can interact with phosphorylates and start a signaling cascade like we looked at with adenocyclase and protein kinase A. So the basic phospholipase C pathway, we activate that phospholipase C with the G protein. We generate DAG and IP3, release calcium from the ER with IP3, and then calcium plus DAG activates protein kinase C. How do you think we shut this pathway off? We hydrolyze that GTP on the G protein, which is activating protein kinase C. We turn that protein kinase C, we stop the downstream parts of that pathway. We reset our G protein. The GDP, it reassociates with gamma and beta subunits and waits for the next signal. So we also talked about in adenocyclase that these signals can be amplified and distributed and interact with multiple signaling pathways to induce multiple cellular responses. One of the ways that this can happen with the phospholipase C pathway, because calcium is involved, calcium not only is involved in activating protein kinase C, but can interact and activate multiple other effector proteins. So one example of that is calmodulin. This is an effector protein. It binds to calcium at each end of the protein, which causes that to change shape. If we look at the protein shape of calmodulin, it's an alpha helix with two hands on either side. Calcium interacts with those two hand structures. And when those hands have those two calciums, they can bind onto the target protein and alter the function of that protein. So here it is stretched out, here it is wrapped around for another protein. So we can distribute that signaling cascade not only through that main phospholipase C pathway, but because calcium is released, that can interact with multiple other effector proteins that can also have an effect on protein activation. Okay. So those are the two pathways we're going to look at for G proteins. Some things you should be familiar with about G protein signaling pathways. As far as our heterotrimeric G protein, there are more than 20 different alpha subunits that have been characterized. Remember, these are the part of the protein that dissociate and go on to interact and start the signaling transduction cascade. There are 5 or so different beta subunits, and 12 or so different gamma subunits that have been described. So it's not a one-to-one association. You get mixing and matching of these components. And the greatest diversity is in that alpha subunit, which is where the GDP to GTP exchange occurs and interacts with downstream pathways. We've talked about two pathways that stimulate some cellular response. You should also be aware that some G proteins, when activated, inhibit cellular responses. So depending on what G proteins are used, you can stimulate or inhibit what's going on in the cell. And we've looked at just 2 specific examples where we are turning on a pathway. And then the final thing about G proteins, remember turning that pathway, once you turn it on, turning it back off is just important. When there are errors in signaling transduction, and you can't turn these pathways off, that generally leads to pretty serious health issues. You need to be able to turn on and turn off that pathway. All right. So that's it for G protein linked pathways. Our final few pathways are enzyme linked. And we're going to talk specifically about pathways that are initiated by the receptor tyrosine kinase. We're calling these enzyme linked pathways because the protein that is receiving the signal from outside the cell is also a kinase itself. So when activated, it itself is doing phosphorylation. So let's look generally at how these tyrosine receptor kinases work, and then we'll look at 2 specific pathways. So our receptors that are tyrosine kinases, here's our membrane. We have on the outside of the cell a receptor for some particular ligand. We have generally a single portion of that protein that passes through the membrane. And then on the other side, we have a portion of that protein that can act as a kinase. And then we have a long tail with a bunch of tyrosines. Remember, tyrosines is one of the 3 amino acids we talked about that these kinases phosphorylate. So in the same protein, we have a kinase and we have a bunch of tyrosines. When this receptor finds a ligand, These receptors tend to aggregate and then phosphorylate the tyrosines on the tails of nearby tyrosine kinase receptors. So because they are phosphorylating molecules that are like themselves, we call this autophosphorylation. And this is why it's enzyme linked. So they receive a signal, they aggregate with other molecules that are like themselves, and then they phosphorylate each other so that their tyrosines on that long tail inside the cell all become phosphorylated. There are multiple proteins that can interact with a phosphorylated tyrosine. We're going to look at two examples of that. We're going to start off looking at something that we just looked at. So the pathway should be very familiar. It just has a different initiating event. So if we look at the types of pathways that are initiated by these tyrosine kinases, there are phospholipase C pathways that can be initiated by these phosphorylated tyrosines. So once these tyrosines are phosphorylated, they interact with phospholipase C, and then everything else from there is a very similar pathway to what we've already seen. Phospholipase C interacts with PIP2, it cleaves it into diacylglycerol and IP3. IP3 opens up calcium channels in the ER. Calcium and DAG activate protein kinase C. All right, so it's the exact same initiation of signal transduction, or I'm sorry, it's the exact same signal transduction pathway, it's a different initiation. Rather than a GTP-bound alpha subunit activating that phospholipase C is activated by interacting with these phosphorylated tyrosines. Phospholipase C, multiple ways that signaling transduction pathway can be initiated. The final pathway, this is it, is the RAS-MAPK pathway. So the RAS-MAPK pathway also interacts with the tyrosine kinase. Tyrosine kinase finds a ligand, they aggregate, they autophosphorylate. One thing that I haven't mentioned yet is how phospholipase C recognizes these phosphorylated tyrosines. So proteins that are interacting with this type of kinase, they have what's called an SH2 domain. And that's simply a domain within the protein that recognizes a phosphorylated tyrosine. So that allows that protein to interact with these kinases once they have been activated. The RAS-MAP pathway involves the RAS protein, which is a common regulator of cell growth. Notice that it is bound to a GDP. So is this RAS protein a G protein? Yes or no? 50-50 chance. What's a G protein? What's their definition of a G protein? Everyone get their answers in? Let's take a look. All right, a little bit of confusion. What's the definition of a G protein? How do you define a G protein? Yes. Guanine nucleotide binding protein. Did that protein bind a guanine nucleotide? Yep, that's what that GDP is, right? So it is a G protein. What's the difference between this G protein and the ones we were just looking at? It's monomeric. So in the GPCR signaling pathway, our G proteins were heterotrimeric. So they're made up of 3 different subunits, which were different. In this particular pathway, we have a monomeric G protein. They're a single unit. But it's still binding our guanine nucleotide, so it's still a G protein. So our RAS protein is bound to a GDP. In order to exchange GDP for GTP and activate that protein, it needs some help. It needs the help of a guanine nucleotide exchange factor. So it needs the protein to do that exchange for it. In this particular pathway, the guanine nucleotide exchange factor is called SOS. SOS, in order to be able to help RAS exchange GDP for GTP, it has to be activated. In order for SOS to be activated, it needs to be interacting with our GRB2. Our GRB2 protein is the protein with that SH2 domain that is going to be activated by binding with our phosphorylated tyrosine. All right, so there are a number of steps involved here. In GPCRs, that G protein coupled receptor received a signal, it bound the G protein, and it initiated that GDP to GTP exchange. In our enzyme pathway, enzyme linked pathway, we have to have a bunch of things activated in order to get to our G protein GDP to GTP exchange. All right, so we sort of worked at it backwards. Let's look at it now the other way around. Once our tyrosine kinase is activated, autophosphorylation, GRB2 that has that SH2 domain can bind to the phosphorylated tyrosines. Then it can associate with SOS. That activates SOS. SOS interacts with RAS and helps RAS with the GDP-GTP exchange. And now RAS is activated. So all of that is our initiation of our G protein signaling cascade. Our receptor tyrosine kinases bind a ligand, autophosphorylate, GRB2 binds to that, SOS binds to that, and then SOS promotes GDP to GTP exchange. So it's a longer initiation set of steps. Once RAS is activated, by that GDP to GTP exchange, now we can start the signaling transduction pathway that's going to lead to a cellular response. RAS interacts with RAF. It phosphorylates RAF, so it's a kinase. Once RAF is phosphorylated, it phosphorylates MEK. So RAF is a kinase. MEK phosphorylates MAPK or MAP kinase. So MEK is a kinase. All right, so we see that signal transduction again. We get activation, phosphorylation of a kinase to activate it, phosphorylation of another kinase to activate it, etc. All right, MAP is short for mitogen activated protein kinase. And these kinases are often effectors of nuclear proteins. So this kinase is in the nucleus, and it is phosphorylating transcription factors. So remember all the way back to transcription, talked about general transcription factors, regulatory transcription factors. So MAPK, by phosphorylating transcription factors, controls gene expression. It regulates gene expression. So here's an example of a pathway that's going from an external signal through into the nucleus to regulate gene expression. So this is a slower cellular response. It has to regulate gene expression in response to some external stimuli. That gene has to be, well, it's either stimulated or inhibited. If it's stimulated, then you have to go through transcription, translation, protein modification in order to see that cellular response. How do we turn off this pathway? The same way we turned off all the other pathways. We hydrolyze that GTP. Right? Turning it off is easy. It's the same in each one of these pathways. Right? In this particular pathway, RAS needed help with its guanine nucleotide exchange. It also needs help hydrolyzing its GTP. So there's one additional protein that is our GTPase activating protein, or GAP. And that GAP hydrolyzes the RAS GTP to GDP, turning off RAS, shutting down the pathway. So, to review, first let's review enzyme-linked pathways. We have our tyrosine kinases, receive a signal, autophosphorylation. Our phospholipase C, some versions of this have that SH2 domain, means they can bind to these phosphorylated tyrosines. It acts just like another phospholipase C. It cleaves PIP2 into DAG and IP3. IP3 binds to an ion channel on the ER. Calcium is released into the cytosol. Calcium and DAG can go on to interact with other proteins, initiating a signal transduction cascade. So that's the first enzyme-linked pathway we looked at. The second pathway, we have GRB2 and SOS. GRB2 has that SH2 domain. They bind to a phosphorylated tyrosine that activates GDP to GTP exchange in RAS. That activates RAS, which interacts through a series of kinases to MAPK. MAPK moves into the nucleus, phosphorylates transcription factors, and affects and regulates gene expression. Sorry, say that again? So RAS and MEK, so once we have RAS GTP activated, it interacts with RAS, which interacts with MEK, which interacts with MAPK. So these two arrows represent that signaling pathway. We end up phosphorylating MAPK, which then can phosphorylate transcription factors. All right. Our three pathways. Vinyl cyclase, phospholipase C in either the GPCR form or the enzyme linked form of activation, and the RAS-MAP. Vinyl cyclase involves a heterotrimeric G protein, activates vinyl cyclase, generates cyclic AMP, activates protein kinase A. Be able to draw that out through to glycogen. We look specifically at the glycogen breakdown pathway in the liver. Also in the heterotrimeric G protein signaling pathways, we looked at phospholipase C. The G protein activates phospholipase C, cleaves PIP2 to DAG and IP3. IP3 releases calcium from the ER. Calcium and DAG activate protein kinase C. Calcium can also interact with other effector proteins. Enzyme links. We look at two pathways through tyrosine kinase receptors. Phospholipase C, same exact sort of signaling cascade to activate a kinase. The initiation is different in that it has an SH2 domain and binds to the phosphorylated tyrosine. All right, so just like we looked at in G protein coupled receptors, just that activation step is different. For the RAS-MAP kinase pathway, we activate RAS through SOS-GRB2. Activated RAS is a monomeric G protein, in contrast to our heterotrimeric G proteins. Activated RAS activates RAF, which is a kinase, which interacts with MEK, which interacts with MAPK, which then phosphorylates transcription factors. With the exam, be able to draw, label, and explain these pathways. Don't forget how they're turned off, and you'll be ready for the final. That is it for this class. No more new material. You have a few minutes of lecture left. If you have any questions you would like to ask me about the final concept. Yeah. So the question is about the RAS-MAP pathway. The question is about why the RAS-MAP pathway is not a heterotrimeric. Why is it enzyme linked and not a trimeric G protein? So it is a G protein pathway, but it is not initiated by a G protein coupled receptor. It's initiated by the receptor itself as an enzyme. So it involves a G protein, but it's not initiated by a G protein coupled receptor. And the G proteins in each pathway are different. And the G protein pathways are heterotrimeric, and the tyrosine kinase are monomeric. Other questions? If you're leaving, please do it quietly so that I can answer questions for anyone who stays. Any additional questions? Now's your chance. Yeah, this So the question is an example of what phospholipase C does. So the GPCR version of phospholipase C is involved in our visual system and initiating the cellular response to photons of light. So that is a GPCR coupled phospholipase C pathway that results in either opening or closing of calcium channels in the cell membrane to initiate a cellular response. Any other questions? Thank you very much for a good semester. I will see you here at 7:30 a.m. Friday, the 3rd.